I would like to thank the organizers for the opportunity to meet with the lupus community and share our work on flipons. Flipons are something you may not have heard about, and they're composed of junk DNA. And so I would like to convince you today that junk DNA is really, really useful and underlies a digital genome that we're only starting to learn about. So codons and flipons are different, and I'll explain how. And then I will go on and describe how flipons regulate immune responses, and also how we can therapeutically target flipons. So as we learned in high school, codons are made from trinucleotide repeats, where, for example, a poly U encodes phenylalanine and a poly G specifies glycine, and other codons tell the translation machinery when to start and stop making proteins. In contrast, flipons encode genetic information by their conformation, by their shape. They act as binary switches because they can be one shape or the other. There are either 0 or 1 elements that can be used to make a digital genome and alter the readout of genetic information into programs. Flipons are formed by repeat sequences, and we will describe those sequences in just a moment. But first of all, what does the flipon look like? So if we start here with the right-handed Watson-Crick bDNA, we can take that and we can band it to form an L-flipon, an architectural element that's recognized by many cell proteins, or we can anneal another strand to the right-handed double-stranded helix and make a third strand triplex, or we can refold the bDNA double helix into a 4-stranded structure called a quadruplex, and we call that a G-flipon. Or else we can take the bases in the right-handed helix and flip them over to form a left-handed helix called Z-DNA. So we can start with repeat sequences, and we can form simple structures, and we can use those to generate complex biology. So what are the repeat sequences that are involved? Well, as you know, most of our genome is repeat, and very little of it is coding. So these repeat sequences that form alternative structures called flipons consist of alternating repeats such as CG-CG or CA-CA, or repeats of the same nucleotides, such as poly-C or poly-G, where C is cytidine, G is guanosine, and A is adenosine. They also can be formed by retrotransposons. These are selfish elements that copy themselves into RNA and then paste themselves back into the genome at any odd place, normally in active genes. Or flipons can be formed by endogenous viruses that have been captured by the genome at some time in history. These flipons can be anywhere within the genome, and by their nature, they are switches. They can change shape from one conformation to the other and alter readout of genetic information at their locus. Because of their effects on phenotype, they are subject to natural selection and are as very much a genetic element as are codons. So flipons require energy to change conformation. It takes work to unfold the right-handed bDNA structure and refold it into a different shape. And where does this energy come from? Well, here's one example. When RNA polymerases transcribe DNA into RNA, they open up the helix and cause strain in their wake. And this strain is sufficient to flip a sequence from right-handed bDNA to left-handed Z-DNA. RNA polymerases aren't the only enzymes that can do this. Helicases can do it as well. And there are other mechanisms for forming Z-DNA within the genome and other mechanisms for forming other flipon types in different contexts. So today we're going to focus on Z-DNA, and as you look at Z-DNA from top to bottom, the helical strand goes from left to right, and that's why it's called Z-DNA. And we're going to talk about its biological functions and how we use genetics to show that it is an element that is active in the genome and has biological importance, both in the immune system and also in causing disease. So here's the structure of the first ever Z-DNA specific protein that we isolated, and it's bound to left-handed Z-DNA. And you can see the backbone of the DNA is zigzag, and that's how Z-DNA got its name, for a zigzag backbone. And you can also see that the Z-alpha domain contacts the Z-DNA helix in a very specific manner, and it's very structure specific, and the binding is very, very tight. Z-alpha binds not only to Z-DNA, but also to Z-RNA. Z-alpha is present in a number of different proteins. The one that we first identified it in is the protein ADA1, and ADA has a deaminase domain, and it edits double-stranded RNA that it recognizes through these double-stranded binding domains. And what it does is it deaminates the adenosine in double-stranded RNA to form inosine. And if this occurs in a codon, it can change the protein that's made by changing the amino acid incorporated. But it also can destabilize double-stranded RNA to give single-stranded RNA. And there are two forms of ADA1, the one that contains Z-alpha, and a shorter form that lacks Z-alpha. And so we can use the difference between these two isoforms to identify what the function of Z-alpha is, using the short form as a control. Z-alpha domain is also present in another protein called Z-DNA binding protein 1. And this has a different functional domain, a rim domain, that enables it to activate a programmed cell death pathway called netroptosis. In other species, the Z-alpha domain is associated with a protein kinase domain, especially in fish, and this enables this fish to turn off translation when they sense the viral infection. So it has a different outcome from in humans. Viruses also have a Z-alpha domain, and they use this protein that they make. It's different for pox viruses like vaccinia that causes smallpox and fish herpes virus. They can use the Z-alpha domain to inhibit the cellular proteins. So E3 Z-alpha will inhibit ADA1 and Z binding protein 1, whereas the fish virus protein ORF112 will inhibit PKZ in fish. So how were we able to identify that Z-DNA is an active element of the genome and has biological functions? We did so by studying two conserved residues in Z-alpha that are key to binding to Z-DNA, an asparagine 173 and a proline 193. We found that variants in these two residues failed to turn off type 1 interferon responses and resulted in a disease called Agouti-Cutaneous syndrome type 6. In this process, we identified three different Z-alpha alleles, a wild type allele, a loss of function allele, and a null allele. And as you all know, we get one allele from our mother, one from our father, and the combination determines phenotype. So with two wild type alleles, shown by the big A, your phenotype is normal. But when you have a loss of function allele, shown by the little a, you have a high interferon response to viral infection. And the frequency of this loss of function allele is about 0.2% worldwide. So we believe it's been under selection because of the advantage it offers in protecting people against viruses. You can also have a situation we have an allele that doesn't make any protein at all, and when combined with a wild type allele, you have about half the amount of active ADAR protein produced. And this produces a syndrome called dyschromatosis symmetrica hereditaria, which is associated with macules and altered pigmentation of the skin, especially in sun-exposed areas. So with two little a's, two loss of function alleles, the phenotype is unknown, although people do have this genotype, they just haven't been characterized yet. So when you have a loss of function allele paired with a null allele, you only get protein expressed from the loss of function allele because there's no protein produced from the other chromosome to cover any effects. So there's a direct one-to-one match between genotype and phenotype. And in this situation, you get Aicardi-Goutieres syndrome, which is characterized by a neurological disease in which you get brain calcification, and normally affected individuals die during childhood. If you have two normal alleles where you make no P150 protein at all, it's believed that you end up with embryonic death, and this is modeled in mouse double knockouts of P150. So how does ADA1 regulate the interferon response? So normally the cell is very sensitive to double-stranded RNA because it's a sign of viral infection, and responds to double-stranded RNA by activating an interferon response to stop the virus. But the problem is that the host also makes double-stranded RNA, particularly from repeat elements such as ALU. So when these repeat elements are encoded on the same strand in opposite orientation, they can base pair to form double-stranded RNA. So if left unchecked, they would activate an interferon response. So the role of ADA is to try and remove these double-stranded RNAs from host genomes so you don't get immune reactions against self. So how does Z-alpha fit into this picture? So it turns out that these ALU elements contain a very highly conserved sequence that forms Z-RNA. This Z-box sequence forms Z-RNA, and that enables ADA1 to recognize those sequences through the Z-alpha domain. What normally happens at the start of the interferon response is there's a protein MDA5 that induces Z formation in these ALU elements, to which P150 Z-alpha binds. So it localizes the enzyme to host transcripts. And as a result of editing, the double-stranded RNA is made single-stranded and the interferon response is inhibited. This is an interesting example where the ALU retrotransposons originally threatened our survival by invading our genome, but now they are used to protect the genome by identifying self-transcripts to stop anti-self immune responses. So ADA1 Z-alpha is part of a larger regulatory scheme. The other Z binding protein in humans is Z-DNA binding protein 1, which activates inflammatory cell death and is triggered by Z-RNA. So Z-RNA is another element recognized by ADA1P50, which inhibits both responses, the interferon response and also the cell death response. Viruses also have their Z-alpha to inhibit these responses. So the viral E3L from smallpox virus inhibits Z-DNA-dependent inflammatory cell death to maintain its virulence. So that's not the only pathology in which ADA1 is involved. Tumors use ADA1 to silence immune responses against them. They inhibit interferon responses, and they use ADA1 to inhibit cell death responses. We found a small molecule that can directly cause the BP1 activation by inducing Z-DNA formation in the tumor stroma. And as a result of the inflammatory cell death, we found that in preclinical models, that immune checkpoint inhibitors work much, much better once you activate Z formation with a small molecule. So this was work done with collaboration with Sid Balachandran from Fox Chase Cancer Center. We also are looking at the role of ADA1 and ZPP1 in inflammatory diseases like lupus erythematosus, but we don't yet have anything to report. But our strong belief is that they play a very important role in such diseases, and hopefully understanding that role will help us develop better therapies for autoimmune diseases. So I'd like to finish just by pointing out that Z-DNA is only one kind of flipon, that there are other flipons that exist that may have different biological roles. For example, the G-Quadruplex is a very stable structure and could provide memory within DNA, within the genome of previous states. Also, elements like the I motif are pH dependent in their formation, and so may be sensors of cell metabolic acidosis, and triplexes and HMG box duplexes also have roles in organizing the structure of chromosomes. Looking at the bigger picture, we have codon wear that represents the semantics of life. We have wet wear that represents the cell machinery of life. And we have flipon wear that represents the adaptability of life. Flipons change the readout of information from the genome. They change the genetic programs run, and they change how inputs are converted to output. By their binary nature, flipons are essential elements of a digital genome, and through their effects on phenotype are subject to natural selection. So as a closing thought, I would like to leave you with the idea that repeat DNA is not junk, but really, really useful for building digital genomes that are highly adaptive to a changing environment. I'd like to thank you for your attention. I would also like to thank my collaborators, Sid Balachandran from Fox Chase Cancer Center and Maria Povzova from the Higher School of Economics in Moscow, who has a group working on analyzing flipons within the data sets generated by the Human Genome Project. So thanks again, and ready to take questions.